Looking at these two coast redwood trees, you might be wondering why the stem of this one is so narrow and why the stem of this one is so massive.
Hi, my name is Alyssa, and I will be starting our presentation on wood formation. So what we'll be going over today will be introduction concepts to wood formation. We'll be talking about the vascular cambium sequence, the cork cambium, axial and ray parenchyma, heartwood versus sapwood, and the importance of wood formation. So going into introduction concepts, secondary growth, which is the growth and girth of a plant from two lateral meristems, happens after sufficient amount of primary growth has occurred. Primary growth is the lengthwise growth of a plant from shoot to root apical meristems, or from shoot and the root apical meristems. It is important to note that secondary growth occurs in dicot and gymnosperm plants. This is because the organization of the stems allows them to form a vascular cambium. This is not the case for monocot plants. Secondary growth produces secondary xylem and secondary phloem. Secondary phloem will contribute to tree bark, while wood is considered to be secondary xylem. So going into vascular cambium and the order of wood formation, we'll start with cell division. So the lateral meristems will form secondary tissue for wood formation. Auxin triggers the beginning of the secondary growth by signaling the development of the vascular cambium. Xylem and phloem will begin to grow out of the bifacial vesicular cambium. Cambium cells form a ring-shaped meristem from vesicular cambium that divide the xylem and phloem of each vascular bundle. Intervesicular cambium, which are parenchyma cells between vascular bundles, will also contribute to this ring. Vascular cambium is made of fusiform cells and ray initials that divide analytically and periclinally. Auxin signal creates pathways that lead to xylem cell differentiation. Divisions that occur periclinally towards the center of the plant form secondary xylem mother cells, and divisions that occur periclinally towards the surface of the plant form secondary phloem mother cells. These divisions increase the diameter of the plant, so the vascular cambium must divide anaclinally to prevent the stem from rupturing. So going into cell expansion, with the help of expansin proteins that bind cellulose and hemicellulose and other primary cell wall elements, xylem mother cells are able to expand to their mature size.
After these xylem cells have reached their mature size, we start to see the secondary cell wall laid down. Sessae, guided by cortical microtubules, synthesize cellulose microfibrils to create the secondary cell wall. Different microtubule patterns help to create the various secondary cell wall shapes that different types of xylem cells require, such as the border pits within tracheids. At this time, the Casparian strip, made of lignin, is also laid down to the endodermis for waterproofing and protection. When wood formation is almost complete, hydrolytic enzymes are released to induce the rupture of vacuoles in xylem cells, which results in their death. The purpose of this is to get all the cellular machinery out of the way so that water can freely flow through the vessel members, creating new wood. In temperate environments with wet and dry seasons, the tracheids and xylem vessel elements vary in diameter based on when they were formed. The spring wood, which forms when water is more available, has larger tracheids and xylem vessel elements. Summer wood is made of smaller tracheids and xylem vessel elements because water is less available to the plant. One annual ring, which is a concentric ring representing the secondary growth that occurs within a year, consists of one ring of spring wood and one ring of summer wood. It's also important to note that as the vascular cambium produces secondary phloem, the primary phloem is being pushed to the plant surface to create an old bark before it's left off. Now we're going to be discussing the cork cambium sequence, and the cork cambium is the second lateral meristem that occurs with wood development. So initially, young plants are protected by an epidermis. As the plant matures, a tougher layer called periderm emerges from the parenchyma cells in the cortex. And that cortex is going to lie outside of the vascular cambium and inside of the epidermis, so it's going to be between those layers. As the cork cambium produces the periderm, it pushes the epidermis to the surface of the plant, and the epidermis eventually sloughs off entirely to complete the emergence of the periderm. The periderm is outside of the vascular cambium, and its purpose is to protect the plant. From outside to inside, it consists of cork, the cork cambium, and phyloderm. Similarly to the vascular cambium, the cork cambium increases the stem diameter with periclinal divisions and prevents rupture through anticlinal divisions. It produces phloem, or cork cells, to the outside and phyloderm to the inside. Phylloderm cells are alive at maturity, whereas outer cork cells are not. These outer cork cells are coated in suberin to protect from water loss and damage. As the diameter grows, the outermost cork cells are sloughed off, but new cork is constantly being replaced to produce it. It's kind of like a snake shedding its skin as it grows.
Next, we will be talking about axial and ray systems. So, the fusiform initials of the vascular cambium make all the secondary xylem and phloem cells of the axial system, which runs up and down the plant. Axial secondary phloem cells can take the form of sieve elements for conduction and signaling, sclerenchyma cells like fibers and scleriids for support, and parenchyma for food storage and translocation. Axial secondary xylem cells can take the form of tracheary elements like tracheids and vessel elements for conduction of water and solutes, fiber for support, and parenchyma for food storage and translocation. The ray initials of the vascular cambium make all of the secondary xylem and phloem cells of the ray system. They make the tracheids and parenchyma of the xylem ray and the parenchyma of the phloem rays. Lastly, we will be talking about heartwood and sapwood and the main differences between the two types. So, xylem rays transport tannins, which are important secondary metabolites that deter predation and work as a fire retardant, to the center of the stem. These tannins darken the center of the stem, creating heartwood, which is composed of dead cells. The lighter colored rings closer to the stem surface that are responsible for conduction comprise the sapwood, which is composed of living cells.
You might be wondering, that's really interesting, but why does it matter for real-life applications? Well, first of all, when a tree is as tall as this redwood, that's a lot of pressure to be putting on the bottom of the tree. So allowing it to grow radially in width allows it to withstand that pressure, as well as the cork cambium building that bark and laying down that suberin layer. It's really important to protect from environmental stressors like water loss during a drought, which we have here in California, and other stressors like insects who might be coming to try to eat it. Additionally, this width is super important because it allows the tree to get super tall, and when you get super tall, it allows you to compete for more light.